So, a very good afternoon to all of you. I welcome you all for the second day of this online training program. And in the second day, we have two speakers for this session, and they will be speaking on the various computational tools that are used in structural bioinformatics, particularly those dealing with the three-dimensional structures of proteins. And this training will incorporate a lot of different tools that are being used for looking into the three-dimensional structures of protein. So our first speaker will be Chiranjeev Sarkar. Chiranjeev is the assistant professor here at Department of Bioinformatics, and Chiranjeev has done his MSc in Bioinformatics from IARI New Delhi. His area of interest include protein modeling, protein-protein interaction, biological network study. So he has more than 3 publications in reputed journal. So now I would request Chiranjeev to take over this presentation and to speak to you. So Chiranjeev.
Thank you, ma'am. Very good afternoon to everyone. And I'm going to talk about protein structure prediction using different computational tool. So I'm sharing my slides. So topic is in silico protein structure prediction. So protein is a macromolecule, so which is a very functional molecule, macromolecule. So why we need to predict a structure of a protein, we have to first understand that. So so structure in any organism, human, plant, in any organism, protein is the most important macromolecule which involve in different biological function. So this is very important to understand if you want to, if we need to understand, if you want to understand the different importance of any protein, the function of any involvement of any protein in a biological process, we need to understand the function of a specific protein, individual protein. So in case of protein, the function of a protein depends on the structure of a protein. That's the tertiary structure of a protein. So there are 3 layers of structure of a protein. So it can be a primary structure, means the amino acid sequence, the simple chain of amino acid. And the second is secondary structure of protein, that is the local conformation primarily stabilized by hydrogen bonding. And the third is tertiary structure. So in case of tertiary structure, there is different possibilities to form the conformation, and which is more we need to identify the more stabilized one, which is in its global free minimum energy. So in case of identifying or predict the protein three-dimensional structure, it is important to identify that stabilized three-dimension for conformation which is involved functionally in different biological processes. So that's why so structure prediction of protein is important biologically in different for different biological processes. So there are different methods for protein structure prediction. Some are experimental methods and some are computational methods. So in experimental methods, usually X-ray crystallography is there, NMR spectroscopy, and there is another one, and the computational process. So in experimental method, there are some drawbacks and disadvantages because of the uh, because of the experimental cost and all this cost effectiveness. Because of all this, sometime is very difficult to predict the protein using this experimental method, this crystallography and NMR spectroscopy. So we need to predict protein structure based on some more cost, less within less cost and less time, which consume more less time. So there are different computational methods also which are available, which are usually nowadays used for protein tertiary structure prediction. These are homology modeling, fault recognition, and ab initio method. And all these are computational method, different computational tool used uh, for this prediction. So these three are the approaches one can use for predict the protein structure computationally. So these are the some steps which involve when we go for computational protein structure prediction. First, we need to start with a protein sequence, and then we need to identify if there is any existing homolog or similar protein structure is available in the database. Means any database where protein structure is stored, like PDB. So with the existing data, with the existing protein database, if there is any available protein structure is there in the protein database, then we need to like you need to analyze, perform the MSA, that is multiple sequence alignment, or simply pairwise alignment between the protein sequence and the protein sequence of the existing structure. So after that, if we find out any homolog in the protein data bank, that is, we need to identify homolog where when we get a similarity of protein, like protein sequence with our, like target protein and the existing protein structure that has stored in a database, that is PTB or somewhere else, some other database. So the similarity percentage should be more than 40%. Means the target sequence and the template sequence should have the identity percentage more than 40%. So if we have a homolog in PDB's database with more than 40% similarity, then we'll go for homology modeling. And if we don't get a proper homolog, that is the similarity percentage more than 40%, then we have to first predict the secondary structure of the protein from the existing data, and then we'll go for if we don't have a similarity less than 40%, that means there is not enough homolog in the database. So in such case, we need to go for apnicium. Now is it audible? Okay, so I was talking about the protein sequence database searching, then multiple sequence alignment, then finding homolog in the protein bank, and when we'll get a identical protein in the protein database with more than 40% similarity, then we'll go for homology modeling. And if we don't get a homolog more than 40% similarity, then we'll go for ab initio modeling. And there is another one, that is fold recognition procedure method. There we need to find out the secondary structure, okay, based on the database search. Okay, then we'll go for fold recognition. So for database searching, we have to perform the okay. so so for So I was talking about the database search using the protein sequence. So I'm going to that part. How can we search database, like protein data bank, that is PDB, where already few high resolution or low resolution or medium resolution protein structure was stored by different organization or scientists. So we can use that database to find out our homolog of our target protein. So for that we'll run blast. So if the audience want to run blast parallelly, they can open in their system blast in blast URL that is available in NCBI. So that is you can open using the link in NCBI. So if you will run blast for identifying homolog, you have to first select the blast P. Blast P means blast for protein. Then we need to just paste one sequence. Okay, 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 okay. Okay, so for database searching, we can use blast tool that is available in NCBI. So this is the site where you can run blast. It is available in NCBI. So first we have to select the database. You have to paste this protein sequence here, or you can also enter an accession number of the protein in which is stored using that accession number if it is stored in the NCBI. If it is not stored in the NCBI, available in the NCBI, then you can also just paste the sequence in FASTA format, and then this database need to be selected as PDB. There are option in the database part: non-redundant protein sequence database, reference proteins, model organisms. So in this list, I will select PDB, that is protein data bank for proteins, where protein structure data are available, because we need to identify that whether their homolog sequence is there in the PDB or not. So we'll run blast using blast P. So after running, it will take few time, and it will show, it is showing the result. So it is showing no result. Wait, let me check. There's some problem, I think. Uh, so using uh, So it is showing no similar search. I think it is there is some problem with the parameter. I'm checking that what is the problem. So maybe there is some problem with the sequence. I don't know. I have to check. So we can do one thing that using this faster sequence or any existing sequence, we can search in the NCBI so that if there is some problem with the in sequence, it can be resolved using NCBI accession ID. So and in the NCBI database, selecting the protein data database, searching with any keyword, you can search with any protein name. So it is the protein which is a blast resistant protein PI54. So there is a list I get from the search result. So using this accession number, I will search in the blast. So this accession number I get from NCBI. Now I'm giving for the search using blast P. Now let's see. Yeah, so I get a list of result sequence. So in the result part, it is showing that there is some description of each result and there are some score given, e-value is given, percentage of identity is given, and there is a accession number which is the PDB accession number 6J5T and chain number is C. So we'll search that accession number in PDB 6J5T. So in PDB, the structure is available because it is stored using this accession number. So using blast P, we can search any faster sequence in PDB. So after that, when we search the sequence against PDB database, we'll get a accession number in the list, and using that accession number, we can open the file in PDB and we can download the PDB file from this PDB database. Here is the option PDB format. I am downloading that sequence PDB file, structural file in the PDB format, and here the in PDB the 3D structure of the protein is visible in this window only. So I'm downloading the PDB file from PDB database, which is similar, which is come as the similar protein or identical protein with 24.72% similarity in the blast P result. So because already I have said, if we try to identify one protein or homolog in PDB of using any protein sequence in FASTA format, it should be the identity percentage should be more than 40%. Then only we can go for a homology modeling. So if we don't get a like an identity percentage more than 40%, then we need to go for ab initio structure prediction or hold recognition method. So here the percentage is, see, in all the search result, the similarity percentage is less than 40%. So for this protein it is not possible to complete the we cannot predict the structure using homology modeling. So that's why we need to go for ab initio prediction. So we go to that later, how we are doing the steps for ab initio structure prediction. Before that, how I like to show you that how to handle the pdb format, pdb file after downloading from pdb database. So this is the pdb file. We can open that. You can, like, anyone can open this using any word file or notepad. You can also open the pdb file using notepad. See, there is a description part of the pdb. So I think it is not visible, so let me share the screen. So this is the notepad pdb file I opened in notepad. So in the first part it is only the description part. So in the protein, what the protein I have, the pdb file I have downloaded, the total number of residue available in this pdb file is more than 20, 40, 24,000, more than 20. It's almost, I think, it's very big protein file. So it is 50,000 residue is there, amino acid residue in this pdb file. So if you see that in each row there is a part, the first part is atom, then each atom there is a number. For each atom there is a number, that is the residue number. Then the name of the residue is asp, that is the triple the amino acid code. Okay, then for other glu, for it is the name of the amino acid, and then the coordinate values are given for each atom of each residue. So now if we want to go for a like homology modeling or a ab initio structure prediction, uh, after using after identifying the homolog from the blast p, we have to identify one template. This is called the template what I downloaded from pdb. So now I'm going back to slide again. Um, so it was the step we are searching database using protein sequence. So after that, I will see that we need to MS conduct, perform the MSA, that is the multiple sequence alignment or the pairwise sequence alignment of the protein of target protein and the protein sequence we obtain from the result after blast search or database search. And then here, in case what in this example demonstration, I have taken the protein sequence of pi54 protein. So there is no homolog available in protein data bank, so that's why the humidity, like an identity percentage, is less. So in for this protein we cannot we cannot go for homology modeling because it is not a good homolog. So we need to go for fold recognition or ab initio modeling. So in this case also, if we want to, if we will go a homolog less than or a protein sequence, protein structure available in protein database less with less than 40% similarity percentage, then the model validity, model quality will not be good. It will be like, we need to will get a worse quality model, a low resolution model. So that's why we can at for demonstration we can do that for like, how to predict the homology model, or how to conduct a homology model or modeling. So for that I'm just showing in using this protein sequence only how to do the homology modeling. দাঁড়ান এক minute. So for homology modeling, we can use Swiss model server, which is available online. So in this server, we need to paste our sequence. So in FASTA format, I have paste the sequence here in Swiss model server, and we can go for simply build model. We can simply apply the build model button, but for other option, there are other option, we can fill up that also. There we can provide a mail ID. So any mail ID you can provide for Swiss model. So from that, just run the server using this build model button, and after few minutes you will get a result. Maybe it will take few minutes. So when you will get a result from this build model, that result that it will definitely follow all the steps for homology modeling. The steps for homology modeling I am discussing in a slide with so these are the steps for homology modeling. In case of norm, like in any stuff, like for any sequence which have an identity percentage more than 40%, so we need to first identify the template structure, obviously using that blast p, and we'll get a template of more than 40% identity percentage. We need to align that protein sequence, template sequence with the target protein sequence, and then we'll get a thread like overall alignment. From that alignment we can make some correction in the alignment, and after that in homology modeling the loop building and the model building was performed, and then the model evaluation step is there after the loop building and model building part. So when we start with a target sequence, obviously at the end of the homology modeling we'll get a model based on template. Maybe if there is no template available, then obviously we don't go for a homology modeling. So if we have a template more than 40% identity percentage, then will definitely get a protein structure of the target protein sequence. Then we have to evaluate the model. That is, in this step, when we'll get a after loop building and the model building step, we'll have to evaluate the model. And if the model is okay, evaluation means that the Ramachandran plot analysis and the energy minimization part of the model of the protein structure of the target sequence. If the energy minimization energy is in the minimized or order, it is in minimized value, then we need not to think about the energy value. We need to think about the other criteria like Ramachandran plot result. So the Ramachandran plot we need to analyze using, there is also a online tool for Ramachandran plot analysis. I'm showing you that. Hmm. So this is Ramachandran plot. So this is Ramachandran plot. So if after getting after after complete the all the steps of homology modeling, that we need not to worry about the steps, because all the steps will be internally, it will be run in the server. So we'll get the output of the protein sequence so in the pdb format. So after we'll get the result of the pdb of the swiss model or homology modeling, then we have to conduct this validation part, evaluation part, using ProCheck, that is available in the as a web tool. So it is freely available. So all the softwares I am showing, all these are freely available. So you need not to worry about the license and purchase. So in this ProCheck SAPes 5 v 5.0. So for in this software web tool, we need to upload the pdb files. It is written here in the instruction part, only pdb files we can submit, not any FASTA file. So in the browse part, just I'm uploading a pdb file. uh, it is a pdb file, it is in my desktop. So for I'm just trying to show how to run uh, pdb file using this ProCheck software, ProCheck online tool. So here is the option run shapes. So when you run the pdb file using this web tool, we'll get a result, a list of documents, and the final result will be shown in this way in the pdf format, or you can open using web tool or any web page also. So it will be also available in the web page, so in the server. So in this from this Ramachandran plot, it is very difficult to understand which residues are in allowed region and which are not in the allowed region. So that's why in the end of this picture, in the below of this picture, the list is given. Okay, manually you need not to identify which is in the favored region and which is in the not allowed region. So in this, for this protein sequence, the residues, it is written, it is the residues in the most favored region, that is number of residues is 51, which is 47.7%, and residues in additional allowed region, that is 38.3%, and residues in generously allowed region, that is 7.5%. So like that, from this list we can see that residues in favored region is very less, because the model I have uploaded in this, that is not refined enough, and that is not I did not done enough energy minimization, so all the residues are not in the favored region. So it from this result, it is, you can see, from this result we can at least understand that one protein which is not stabilized properly or refined properly, the in for such protein the residues in favored region will be less. So we need to refine that protein again and again. We need to check using this Ramachandran plot. So until and unless we get a good number of favored residues in favored region, that is almost usually it is theoretically it is said that 98 more than 98% residue should be in the favored region. So if it is close to 98% or more than 90%, that is good. So until and unless we get a 99 more than 90% region residues in the favored region, we need to repeatedly perform this operation. Run means once after checking this Ramachandran plot, we need to again go for a loop refinement or side chain refinement. So we need to find out, definitely it's very difficult and until and unless one by one using side chain refinement, loop refinement is not easy. So before that I'm just showing you, it is already the result. The protein sequence have submitted in the Swiss model. It is there in the result has been given. So in this window it is showing the protein sequence predicted using this server and the template they have used by default. If you don't mention any template, there is the option template. So if you don't mention any template, by default it will identify any template. See that identity percent of the template is 23.96%. So so the model quality will not be good. Definitely it is template is not homolog and it is less than 40% similarity. So we need to refine it, because if you don't refine it properly, the side chain and the loop will not be modeled properly, so the quality of the structure will not be good. So the pdb file we can download from here, the pdb file of the submitted structure. So the pdb file you can download, you can later you can try this, download the pdb file, and then you have to check in Ramachandran plot, and then if it is not showing 98% residue or more than 90% residue in the favored region, then you have to you have to again go for mod loop refinement. There is a software loop for loop refinement, is mod refiner. It is available in this site Jang Lab. So there are different tools are available in this site also. So it is one of them, mod refiner. It is used for um, loop refinement or side chain refinement. So there is demonstration how can, which the theory is given. So I'm not reading that theory actually. I'm just trying to demonstrate how can you submit or upload a pdb file in this tool, and what will be the result. The result from we'll get from this mod refiner, how to deal with that result. Okay, so structure file of the target structure file, uploading option is here. So I'm uploading the file. I'm uploading the same file example, that is a pdb file. So you can simply run using this. So you have to provide a mail id obviously. So it must be, I think, it is not mandatory. You have to just send write a mail id and you can run, and the result, when the run will complete, the result will be sent in your mail id or a link will be sent. So this is the mod refiner, mod refiner tool, where it is showing the initial model, the model I have submitted now. The model of, this model is showing now, and after the run complete, it will show the result in this way. There is a example um, file, the how it is showing the output file. Once if you try this, you will get a result like this. you have to submit the pdb file. Then you have to, you not to with this default parameter, there is a parameter list in the mod refiner. So you can run the mod refiner using default parameter, and you will get a reference refined model in the output window. So here it is, three option is there: initial model, what the model you have submitted, then the reference model, using that reference model it is refine the model, and then it is the final refined model is showing here. So you can download the model, and it is also so download in pdb format. So after refinement, again you have to check the quality of the model using this Ramachandran plot, and if it is not showing like expected result, then like allowed like residues in the favored region, that should be more than 90%. If it is not showing that, then if you have to again run mod refiner, and then like in it you have to run this in a repetitive way, like until and unless you get a proper expected result in the Ramachandran plot. So after that, after done with this Ramachandran plot and mod refinement, once you get number of residues in the favored region is expected more than 90%, so it is obvious. So now your model is properly refined and loop has been modeled properly. Now you have to think, like at the end of all this, the thing come for homology modeling, protein structure prediction and protein modeling, that is energy minimization. So there are other tool which are which you can standalone tool which you can download and run in your system. So I am just demonstrate using online tool which is freely available. So this is Yasara minimization server, where you can minimize your minimize the energy uh, of the protein. You have to provide the mail address here also, and you have to enter a PDB ID, means the PDB existing PDB protein structure in the PDB file, or you can upload your own PDB file. So it will show, obviously it is also give you the result by mail. So you will get the result later in the mail ID. So it will obviously it will show in the output window, there will be only one protein file in the pdb format in the minimized energy, and it will calculate the energy, it will show the energy level of the protein. So you can just go, you can just run this, and after you will run, you will get a pdb file, you can download that, and after that your model is ready. So you have to be careful about the steps. Once you have submitted, you have for homology you have to once I'm showing the steps again, once again, for homology modeling. So first you have to identify a template using blast search, blast database. Then you can correct the template structure and the protein target protein sequence, protein target protein and the template protein sequence, and then you can simply go for homology modeling. If the percentage template the identity percentage with the template is more than 40%, so there is no problem, you can simply submit your protein sequence in the Swiss model server, and then you the output, you have to evaluate the model, and you have to start with Ramachandran plot. First you have to run the Ramachandran plot, then you have to check the residues in the Ramachandra favored region in the Ramachandran plot is more than 90% or not. If it is not, then you have to run the mod refiner, and then again you have to check the Ramachandran plot until and unless you get a the residues, 90% of the residues or more than 90% of the residues in the allowed region or the favored region. So after evaluation or mod refinement and proper validation using Ramachandran plot, you can go for energy minimization, and once you have done the energy minimization, your model is ready using homology modeling. So after that, for other like functional analysis or for other uh, task, you can use your protein now. So that is homology modeling. So these are some links, and so now I'm going to discuss about the ab initio protein structure prediction. So in case of ab initio protein structure modeling, the thing is, when you don't get a template more than 40% identity with your existing protein structure in the PDB, then you have to go for ab initio protein structure prediction, because in ab initio protein structure it denote the algorithm of ab initio protein structure modeling in the like available in web tool and different as a software. They don't follow, they don't need a template, and so they can simply predict, they can model the protein using the physics and knowledge based potentials, means the physiochemical properties, hydrogen bonding, and the chemical properties. Based on this, they in ab initio protein modeling, first conformational search has been conducted, and then when we perform a conformational search, means for one protein sequence we will first generate a huge number of conformation, and for this, in ab initio protein structure prediction, it takes more time than homology modeling, because for conformation search and more conformation you generate for your structure prediction, then hello, hello. So more confirmation you generate for your confirmation search, the more accurate protein model you will get from at the end of the structure prediction. So as it is based on the physiochemical properties and physics and knowledge based procedure of protein modeling, so confirmation search is very important, and from this confirmation search, a low resolution model I will get from this confirmation search, and from that, after from that low resolution model, again you have to follow the steps what I described for homology modeling. You have to start that evaluation procedure from this low resolution model part. You will get the resolution model from this confirmation search, then you have to refine the model at atomic level. In ab initio protein modeling, the softwares and tools available in database or in web, that's all are follow this algorithm. So maybe the conformational search algorithm is different. In some case it is MCMC algorithm, for some case it is a genetic algorithm, but the basic algorithm is based on this thing. The conformational search will be first generate search will be search will conduct based on some confirmation generation, and it should be based on the length of the protein sequence. If it is large protein, then the confirmation of the protein should be huge. For at least 10,000 or 20,000 confirmation can be considered for confirmation search. Then loop refinement or model refinement part is there. Then again, energy minimization you have to conduct, and the end of the energy minimization, after energy minimization you have to follow the model validation part, and which from that after model validation completed, then you'll get the final model based on ab initio protein modeling. So this is the steps for ab initio modeling. So for ab initio protein modeling, the software available, that is iTester. There is a software which is available, that is freely available. So one can use iTester for ab initio protein structure prediction. So in this protein structure, in this server also, you have to paste your protein sequence to get the protein model using ab initio protein structure. So there is a restriction, you can only add sequence, protein sequence in FASTA format. That should be length, that's the length, there is a restriction, that should be more than 10 amino acid residue, and the maximum length should be 1500 amino acid residue. It should not exceed the limit of amino acid residue more than 1500. So I'm pasting the protein sequence here. This is the ID, it is not the protein sequence. Wait, in the from the NCBI, there, just let me find out some protein sequence. So any protein you can any protein you can search in the NCBI. So from that sequence, for the sequence, just take any sequence. I'm taking a small sequence so that we can get the result as soon as possible. So it is 154 amino acid, the length is very less. In iTester, just paste the sequence, or you can also upload a sequence file. If the sequence length is large, then you can also upload the protein sequence file from your system computer and run the iTester using this run button. And there are some option. So there are some option which is the parameter. So if you don't want to, if you want to add some parameter for complete the submit the run, you can add using this options. So otherwise, by default, if you want to run, you can simply run the iTester after adding the protein sequence. So okay, so I did not add the mail address. You are still in this case also, you have to add the mail ID. So okay, okay, so I need to provide the password also. Which, so once you submit the putting all the information uh, using main, you have to, in all the online tool, you have to add the mail ID, one mail ID, that can be a Gmail ID or any institutional mail ID, or you have to, for this iTester, you have to create a password or login, and you need to provide that password in this, and you can simply run iTester, and then the result file, it will take some time, maybe 1 day or 2 day it can take, but uh, for some protein it takes 7 days also, but you will get the update from the server page regularly, that whether what is the status of your protein sequence, because it takes time, because there is the option of port confirmation search, and for huge number of confirmation search, it takes more time. The more the length of the protein you will consider, then it will take more time. So at the output window you will get a result like this. The final model will be, it will provide few model, highest five, best five model it will provide. So you can download the model from this download link, and it is visible, the protein structure is also visible. So you can also download the any, all the five pdb file, and you can check the manually one by one using mod refiner. The mod refiner already I have show how to run mod refiner. You can simply download all the protein file from protein data, all the pdb file from this, and check the quality of the protein using mod refiner. And again you have to follow the pattern of, you have to follow the steps of checking, validating the protein model using Ramachandran plot. And again, if it is not satisfactory, if it is not showing the satisfactory result, then it will, you have to again refine the model. And at the end, after all these, you have to minimize the energy of the model, refined model. So this is all about the ab initio protein structure prediction. So those who are in live, you can try if you face any problem. So this is my end of my slide. So if there is any question answer session, so you can ask some question.
Has completed his slides. So I think we can move to the question answer session. So if you have any question, you can put your question across in the comment section, and then we can answer your question. There are some questions I will put in the slide so that Chiranjeev can answer. And if there is any new question, please put them in the comment section of the YouTube. Yes, Chiranjeev, there is a question. Put on your mic.
Okay. The file format in Swiss model should be FASTA format, and you can also definitely in notepad you have to open the Swiss the template file, and then you can set, you can simply download the pdb file from your from the pdb database, and simply you can write that accession id, or you can upload that pdb file as a template. Or you can also set your template file using edit file. You can just edit the pdb file in notepad also. Ramachandran plot for used for to validate the protein. Actually, there are different criteria for validating the protein, but for for easy understanding, you can simply you can just validate the protein using Ramachandran plot. So there is, I shown the software that is saves, SAVES, saves ProCheck. The software that is available online, so you can use that for getting Ramachandran plot. I can provide their slide, because actually there is a limitation of time in the, so next speaker is waiting, so I can send that slide in a flow chart, and I can provide that slide with this live also. I can provide later in the YouTube video. Because see, there is no actually ITSR provide you 5 based model, and among them, based on your criteria, suppose you are predicting one protein, you have considered one protein sequence, you have some information based about that protein sequence, the type of the function of the protein, or type of the protein, which type of the protein like what is the biological processes is involved. So based on that, you have to check manually using mod refiner, energy minimization. So using different criteria, because it is a prediction, it cannot be 100% accurate. So it provide 5 based model, and among this 5 based model, you have to apply your theory also, that which biological process or which basic information you have regarding this protein. If you don't have any information, then also you can consider that mod refiner and pro-check result, means energy minimization, then mod refinement. Based on that also you can select the best one among these 5 model.
That's all, I think. And if you have any question, so you can. Yes. So if you have any question, so please, we can. You can put them as comments, and we will try to get back to you. So moving on, our next speaker is here, and so our next speaker is Dr. Amita Barik, and I would like to introduce Amita before you. Amita, Dr. Amita Barik is currently working as assistant professor in Department of Biotechnology in IIT Durgapur. She has completed her PhD from IIT Kharagpur, and she has worked in various institutes like NICED and Virginia Commonwealth University of Asia. She has lots of experience in protein structure predictions, then RNA bioinformatics, and her current topic is intrinsic disorder proteins and their prediction. She has many reputed publications in journals like Nucleic Acid Research, RNA Biology, and JBAC. So I would request Samita to take over, and maybe she can give you
Uh,
her.
Dr. Thakur, am I audible?
Yes, yes,
Hello?
yes, you are.
Hello? Am I audible?
Yes, you are.
Actually, I'm not able to hear you properly.
Okay, so you are audible. You can.
Can you hear me? Hello?
Yes, we can hear you. Yes, we can hear you. Amita.
Okay. So should I start then?
Yes, yes.
Okay. Okay, good. Okay. Okay. Thank you for the introduction, Dr. Thakur, and good evening to one and all. So, first of all, I'd like to appreciate your in this initiation of organizing this kind of workshop for the students, which I believe would be very much useful, particularly at this stage of this pandemic where the students are have limited access to the experimental labs. And the way bioinformatics and computational biology is expanding, I believe that understanding this kind of computational tools will be very much useful for the student community. Oh, my voice is not audible. I got this message. Okay, let me remove this. Hello, can you hear me?
Yes, yes,
Am I audible now?
yes.
Okay, okay, good. I got the message. Yeah. So when Dr. Thakur approached me regarding this workshop, I was a bit skeptical what to talk and all. I was thinking of initially, I thought that I would be giving a talk on RNAs, which happens to be my research area from the past decade. And then I thought that why not to give a idea on something new, recent one, which the students can be using, it will be useful for them, and they can know something better on this new topic. So I am sharing my screen. Yes, the screen. So is the screen visible? Can you hear me, anyone? I'm not able to hear anything. Okay, good. So yeah, I will be discussing today about the intrinsically disordered proteins, which happens to be a kind of recent research area. And since you now learned about proteins in details, I believe I joined the talk a little late, and I heard about Dr. Sarkar speaking about something on ITES. I get some prediction models and PDBs and all. So without further delaying, I'll start my presentation on IDPs, intrinsically disordered proteins. So yeah. Good. So what exactly we know about proteins is that we have been hearing about proteins since childhood days, when the mother used to say, "But I eat protein, eat meat, fish, or egg, you will have proteins," and all that. When we went to the school level, we found out that the proteins are large macromolecules or the building blocks, which are again formed of different monomers such as amino acids. And we also learned about amino acid structures, how this amino acid looks like. There are 20 different kind of amino acids, and the amino acids are joined together with the help of peptide bond to form proteins. We have also learned about the protein structure, hierarchical structure, how the primary structure looks like, how the secondary structure, the alpha helices, beta sheets, and then they form the tertiary structure, and finally the quaternary structures. Also, in the books, we have learned about the protein folding mechanisms, the protein synthesis, how it happens in the ribosome, the protein translation machinery, everything. In fact, we also know about this Ramchandran plot that we are discussing few minutes ago with Dr. Sarkar, when he was telling about what are the structures that are actually happening in this proteins, allowed in the protein regions. So these all things we know about the proteins. Apart from this, what we also know is the protein structure function relationship. That is what we have been taught from our bachelor's days, master's day, what do you say? Okay, since our bachelor's day or master's day. So we say that the protein function is actually depends on the three-dimensional structure of the protein. So a protein needs to have a proper 3D structure so to carry out its function. That is the concept, that is the paradigm that we have been learned, in the textbooks or whatever. And these all were there in this books, the famous books of Lenningers or Strayer, whatever you say, whatever the people study in for the biochemists, these are the Bibles, right? So this all things were there in my mind also, unless until I came up to stumble upon this structure known as IDPs. Now, as it is said that biology is the science of exceptions, this thing actually, this particular sentence actually was there in our class 12 CBSE book. If you are from CBSE medium, you can just go read it. I don't know now they have changed the syllabus or not. But when our biology ma'am taught this, biology is the science of exceptions, as a student, I was like wondering what does that mean. We just read it, we wrote the answers, we just mugged up something and gave the answers. But now when we are coming into this research field, we are looking into this kind of things that are weird, things that are coming up. We are really interested in that biology is really the science of exceptions.
And thing actually this thing actually gave me the idea on this IDPs came up to me when first of all I went to this professor's lab during my postdoctoral research, Professor Lucas Kirkan. He was the one who actually gave me the idea of IDPs, and I worked with him on this project on IDPs. And there he actually gave me an opportunity to meet this professor, Professor A.K. Dankar, who is the pioneer of IDPs. You look into IDPs, search in Google search, Google Scholar, ResearchGate, look into n number of papers, you will definitely find this person's name in all the IDPs. And you know, whatever papers or books you read and you gain knowledge, it is always beneficial. But the way you talk to a person and you get to know the knowledge from that person's personal experience is something very interesting. That is what we had a long discussion with Dr. Professor Dunker. He was in our lab for a couple of hours, and he was discussing with us different stories, how he found out about IDPs, what are the different qualities and all that. And it actually gave me an interest into that. So that is how I also started learning about IDPs. And I must say that, I mean, it is really very interesting to know about IDPs. And I'm recently started the work on IDPs in my lab. I have a small lab, and to be fair and frank, I'm not very much very much deeper into IDPs. I do not know much. I'm still in the exploring stage, so I will try my best to answer the questions whatever you have, and I will also share my knowledge whatever I have on IDPs. Okay, great. So today's contents will be basically divided into two parts. I will first discussing on intrinsically disordered proteins, the IDPs, and then I will be discussing on the computational tools in IDPs. So basically this workshop is on computational tools, so we'll be mostly focusing on the tools, and I will be giving a brief description about the IDPs, what exactly are IDPs. So the first question, what are IDPs and IDRs? So intrinsic disordered proteins, or commonly known as IDPs, and the intrinsically disordered protein regions, or the IDRs, these particular structures are lacking stable tertiary structure. So these proteins do not have a proper three-dimensional structure, and they form dynamic conformational ensembles under physiological conditions. So they keep changing their shapes. That is what the definition of IDPs is. Now, why exactly the word intrinsic? So intrinsically actually indicates it's a sequence dependent characteristics of this proteins. Okay, so why I discuss that, I will discuss coming up in this slide. And this kind of proteins can be fully or partially disordered, but they are quite abundant in both prokaryotes and eukaryotes. Of course, they are more abundant, more found in eukaryotes because of the multicellular organism complexity. And in the context of protein, these are known as the dark region, the dark proteome. They've been given this name by the research community. Now, why are IDPs important? Now, IDPs, although they do not form a stable structure, they play a very vital role in different cellular functions, starting from cell signaling to molecular recognition, translation, transcription, apoptosis, immune response, viral life cycle, and many others. And in fact, they are mostly contributing to human diseases as well. So nowadays, they are also considered as very attractive new class of targets for the drug discovery process. Remember, that is why IDPs are important. Now, let me share two examples of IDPs with you. So this is a breast oncoprotein, LMO4, which interacts with various tandem LIM domain containing proteins that are mediated by the disordered regions. Then there is another very interesting protein, the tumor suppressor protein. I guess all of you are aware of the BRCA1. So this particular protein also binds with multiple protein and DNA with its disordered region, which is actually around 1500 amino acid residues. It has a disorder region which is actually binding to the multiple proteins and DNA regions, and then it is forming a stable complex. Now, IDPs actually, there are certain there are certain notes on this. IDPs have certain binding regions. There are certain short linear motifs which are popularly known as SLIMs. These are actually short in the sense that the average size is around 6 to 7 residues, and they are very much enriched in ideas. So you remember a big chunk of protein sequence we are having, and you are getting small regions or SLIMs around the proteins, and which are actually disorders. Then there comes the region known as MOFs, the molecular recognition features. These are quite a little bigger, longer than the SLIMs, usually around 30 residues, can be up to 70 as well. But this kind of MOFs actually undergo disorder to order transition upon binding to the partners. It might be protein or DNA or RNA or any other linker. Okay. So this is something about the IDPs, a general view on the IDPs for people who do not know about IDPs. So this is a picture that I took from one of the reviews in 2014. It came up. So this is actually has the discovery of IDPs has actually challenged the established existing paradigm, the structure function relationship, which says that a particular structure, a protein should have a particular structure to carry out the function. Now, this concept of disorder has actually changed because the disorder do not have a 3D structure, yet they are functioning. So that is why people are up to this, finding out new things about IDPs. Many people are carrying out experiments, and many theoretical people are using also computational tools and developing web servers, databases on IDPs. Okay. Yeah. So the most important question that comes to our mind, that why actually IDPs do not fold into 3D structure? What is the reason behind it? So amino acids, because the proteins are made up of amino acids. So the composition of actually amino acid composition, actually it is the key factor which determines whether a protein will fold or remain unfold. So there are certain patterns of the hydrophobic and the hydrophilic groups which is actually determining how a protein is folding into a three-dimensional structures. So basically there are certain kind of amino acids in the IDPs because of which it is actually not able to fold into a stable 3D structure. Yeah. So if you look into the amino acid composition in IDPs, what it has been found through research, so this kind of proteins are actually having a high proportion of polar and charged amino acid. Polar and charged amino acids such as glutamine, serine, proline, glutamic acid, and lysine. And they have a low amount of the bulky hydrophobic amino acid, because when they are forming a 3D structure, if you know the protein structure, how it forms, the hydrophilic are on the surface, hydrophobic from the core and all. So hydrophobic amino acids are having low proportions, such as the valine, leucine, isoleucine, methionine, phenylalanine, tryptophan, and tyrosine. This kind of amino acid, if we look into the composition of this amino acids in IDP, you can see that this kind of amino acids are quite few in the IDPs as compared to the polar and charged amino acids. And that is the reason behind the lack of three-dimensional structures of IDPs in the context of reference science. Yeah, so when we are discussing about computational methods and computational tools, I will be discussing about the tools, but let me discuss something about the databases that are meant for the IDPs. So there are many databases actually, so which are storing this kind of IDPs for, so that the student community or the research community can be benefited from this. So these are some of the databases that I have listed. The first one is Disprot and Ideal. So basically, Disprot and Ideal are the two databases which are actually storing the all the experimentally validated disorder regions or disorder proteins, whatever is had been reported in the literature or the scientific reviews or whatever. And these two databases, these are also very nicely curated, the MobiDB and the D2P2. But this actually also have the predicted disordered regions. So apart from the experimental ones, they also keep the predictions as well. They are storing those things. So I'm just showing you a screenshot because I thought that going back to all the databases one by one, and it will be difficult and it will take some time because of internet connectivity. So this is the screenshot of this prod database. Recently they have updated it to the version 8, and now currently they are having around 1600 something like data or IDPs. So as you can look, they have this home page where they are showing that they are manually created from the literature and this annotations. If you look, go to the browse and release notes, you can get all the informations. You can search the database. You can, from the download option, you can download the entire Discord database, most in both in FASTA format or JSON format, whatever you used to have. And then you have different organisms also. Based on the different organisms also, the IDPs have been classified. You can, if you are interested in, suppose let's say Arabidopsis thaliana, you can go and have a look into that. There are 34 IDPs reported from the Arabidopsis thaliana, and more in the, as I said, that multicellular complex organism have more number of IDPs. So in humans we have around 591 that has been reported over here. So the number of proteins are around 1.6k, and then there are something known as ambiguous and non-ambiguous, which actually have a, like, people are not sure whether these are IDPs or not, so they have also kept that also into account. So proteins and the regions, also the residues information and the disordered content, everything they have given in a very statistics way. You can search here in the search box and go, or you can also go into download or into the browse part, and you can find out the different IDPs. Now there is another database I was talking about, Ideal. Ideal also has the experimentally validated IDPs. So you can also go here and search for the search option is there, and then there are different options where you can go the browse, search, download, and statistics history again. So these are the two databases which actually people are using the most for developing different tools which they can get the information, the authentic information. Now coming to the other third database, known as MobiDB. So I was telling that MobiDB is actually having both experimental ones as well as the predictions. So you can look into this, search the MobiDB database. You can have, this is the URL. I actually gave the URL of everything here in the top. You can just look, or else you can simply search in Google MobiDB database. It will come up and you can just find out. So they have actually curated so many proteins, around 90 million proteins are there in the MobiDB database, 90 millions. So these are actually having both the experimental ones as well as the predictions. Similarly, we have also with us D2P2, which is also a database of disordered protein predictions. So we have a number of predictions from the different computational web servers and computational tools, and they have been properly stored in this database known as D2P2. You can go to this URL and find out about D2P2 more. Now, coming to our main topic, computational prediction of IDPs or IDPs. Now, why do we need to predict the IDPs? Since we know that IDPs are having a lot of importance in cellular processes, they have number of functions. It is very much challenging, but it is very much challenging by the experimental people to get the IDRs or the IDPs annotations. So that is why a number of computational tools have been developed which are making our task much easier. And also because the disorder can actually be predicted from the sequence with fair good accuracy, and also there are very good large databases as we discussed where we have all the informations of these proteins. So it is very reasonable that we can develop some computational tools or approaches so that we can know for this potential relationship between the functions and the disorder. And that is why different people are actually developing different computational tools for prediction of IDPs and the IDRs. Yeah. So if you look into the literature, you will find a number of predictors on IDPs and IDRs. So if you generalize and classify this predictors, so you can, there are three basic classes on which you can classify these predictors. The first one is the based on the physicochemical properties. You can take the sequence, look into the physicochemical properties of the amino acids and check for different features like entropy, sequence conservation, evaluation conservation, then the surface area and all that. And you can, based on those features, you can always go for a developing a predictor to identify or the classify the IDs. Now there are certain other kind of predictors which are actually based on machine learning approaches. So as you know, machine learning and AI are now in high demand, and people are trying to use these approaches very well into these predictions. So these are also different kind of predictors which are based on the ML and AI. And then the third class of predictors, which are actually a meta approach, which actually combines different multiple predictors. So it is a kind of combination approach where they are trying to use different predictors, and they are making, developing a combining those predictors and finding a final one, different approaches. So here I tried to summarize all the kind of servers there. I might have missed many because there are actually many web servers and the tools. So here I tried to summarize some of the servers that I came across from the literature. So these are the methods, the predictors, and here the type means what kind of type it is available. So you can see that WS means web server. So it will be like kind of link will be given, you can go for that, and it will web server, it will be having a box like enter your sequence or something like that, and you can submit it and you can get an output results. And the SC stands for standalone program, in the sense that this kind of program softwares or the computational tools have been developed, which have been coming in a kind of package. You need to download those things, you can install it in your PC or computer, and then you can use it. So mostly the standalone versions are mostly in for the Linux version, Linux operating system. And NA stands for that nothing is available. It is not. It was there. I mean, it is there for the in the paper, in the research, but they did not provide any web server. They did not included in the web server or any standalone program. So you can see that summarize that there are very few web servers, so, and also standalone programs, but there are many also. I just did not put everything into this, and this URL I have also set here. You can just look into this, because many times what happens, sometimes the Google search fails, where you write something and it directs something. Anchor, if you suppose write anchor on Google, it will show you something different. So you need to write anchor, predictors and all that. So better you search this, put this URL and get the server itself to get the your task done for the predictions. Okay. Yeah. So among this, I did not write about one nice predictor, which is IUPRED-2A. So actually, IUPRED-2A, this is a nice tool for finding out the prediction of this IDs, and this is a very recent one, which is actually giving a very authentic and good accuracy. And I thought that I will be putting a screenshot for this, because I will be showing the other tools. It might take some time, because it takes some time. So this is the prediction. I will be showing you also how it is works on, but I showed you the screenshot here. So this is the homepage of IUPRED-2A. This is the URL, and here you can see, here it asks for a Swiss prot or a Tremble identifier or the SSN number, where you can just put it into that, or you can, as the your email ID, you can just give it here, or you can also upload a file which contains the FASTA sequences. and you will once upload it, it will be giving you some results. I'm showing you how. Also you can paste the amino sequence. Either you can upload a file or paste the sequence, and this will give you a result something like this. Okay, so so this will be showing you the position of the amino acid sequence, the actual protein sequence. So it will starting from the first, second, and third, and all that. So suppose in the 50th position, this red line and the blue lines, the red line is for the IUPACT2 predictor and anchor 2 is shown in the blue. These are two different predictors which actually have been merged into IUPACT2A. Initially it was IUPACT2, now they included anchor. This is another predictor, so they have made this IUPACT2A. So you must be seeing that what is this lines and all that. So basically in any predictor for IDPs, you have two different scores. One is a binary score, one is a propensity score. So binary and propensity. So propensity is calculated in terms of like numbers, so regions. Like it will be starting from like say 0 to 1 to what will be the chances of this particular position in the protein sequence of being a disorder or not. Let's say it has 0.1, 0.2, 0.3 and so on. So we have actually a cutoff range. Let's say here they have given a cutoff of 0.05. Okay. So here you can see that the regions, the sequence, the protein positions which are having a propensity score more than 0.5. Suppose this region, this one, which is around, this one must be around 30 residue. The 30 residue is having a propensity of more than 0.5. So it has been classified into a disordered residue. Like it has the chances of being into the disorder region. Similarly, this all regions, this part all comes have a high chance of forming a disordered regions, and these are the ordered part. Okay, below the score. This is the all about propensity score. Now coming to what is actually binary. Now binary means, just the name suggests, binary means 0 or 1, either this or that. So based on this propensity scores, which we are classifying a residue to be disordered or not, like disordered or ordered. Okay, so if it crosses more than 0.5, we classify it as a disorder, D, let's say, and if it is less than 0.5, we are classifying it as an ordered. Okay, so there's a cutoff range of this propensity. So first we are having the propensity scores, and the based on that cutoff threshold part, which is actually here they have taken 0.5 based on again literatures, and then the more and more experiments, though, so that they have discussed in 0.5, and based on that threshold value, they have divided this particular residue into a binary. Values have been given. So if you look into, this is the result page, the graphic page of IUPACT2, how this looks like, and then they have also an option of this download results. You can click into this and it will be giving you a text file like this, which you can always download and use it. So here what they have shown, you can see this, the first two lines are for the paper's name and the author's name, and here this in the first column is the position. So 1, 2, 3, 4 and so till 32 I have just taken the screenshot, and the amino acid name. So in the first question, M is there, the single letter code of the amino acid. So M is for methionine, so methionine is there. And here in the third column is the IUPACT score, and the fourth column stands for the anchor score. The IUPACT score is around 0.08 and the anchor score is around 0.3129. So this way each and every position has been given assigned a score. Okay, so based on this scores, you can now say that this particular residue has the tendency to form a disorder or not. So you can see, look into this, if it is visible in your screen or not. So here you can see 0.0, 0.1, point, something like that, and here something on 26 residue, as I was showing. 26 residue is having around 0.6, and 27 also 0.6, 28 also 0.622, and 29, 30 these are having. Suppose here you can see the 26 residue I was telling now 0.6 something. So these peak. You can look into this, and they are having a very high score. That means that they have the chances of being a disordered region. So there are many other tools, many other web servers like Spot is there, iPred is there, and for the protein and RNA binding, there are DMR Pred, DSORDP bind, and so many things. So what today I can show you is that in my stay in VCU in Professor Kuran's lab, what we did at that, why not to combine all these classifiers, all these predictors into one centralized web server so that the research community can be benefited. And see here, if this you look into this output file, it is difficult to parse for the common biologist people. For the computational people, write a code, write a script, you can definitely go and find it out. But what happens for the biologist, common biologist, experimental people, if you know bana banaya thal kind of thing, if you give it in a very nice proper format, they will be like using it, that will be easy for them to use. So what we did, we tried to take out all the different fast solvers that are available in the research literature, which are actually new and also very fast and are also giving good accuracies. And we contacted those authors, we tried to communicate those people and tried to get their programs and tried to keep it to a one centralized part. So that is how we made this web server known as Depictor, where I can be showing you how to use this Depictor. And for that, I'll think I should better go to this web server to show this. So this is a screenshot of the home page of how the Depictor looks like. Okay, so going back, I'm just stop sharing, and I can go back and I can share one screen, which is just
Just share your screen again.
yeah, I'm showing that. Yeah. Is this visible now?
Yes, it's
Yeah. Okay, great. So this is the Depictor web server homepage, how this looks like. So you have different links like tutorial, method, references, and all, whatever you wish to take, you can find it out. So I'm just discussing how to use this Depictor. So this server is actually meant for prediction of IDRs, intrinsically disordered regions, and also their molecular functions and their patterns. So the first step is you need to upload a protein sequence. You can choose a file or you can paste it directly over here. But be specific that it should be in FASTA format sequence. And how does FASTA format? I believe everyone knows it starts with a greater than symbol with a header file followed by the sequence. Okay, so you can also have for the beginners, they have also shown how the FASTA format looks like. Okay, the second option is you need to provide your email address, which is again optional, but I recommend that you should actually put it so that if by chance you lose your results somehow, so you will be given a link to this email address where you can anytime access that link and you can retrieve your results at any point of time. But the results are stored for actually 1 month. After that, they are deleted from the web server. Sorry, the server. And then we have number of predictors over here. So the third step is so by default I have chosen, we have selected all this. So by default it comes, all the predictors have been selected. So these are the disordered residue predictors. The best one we have been chosen for, suppose IUPRED L, IUPRED S, and spot disorder. What I was showing you was IUPRED 2A, which is actually combining IUPRED L and another anchor. So anchor is basically a predictor for the protein binding residue. So we have selected for the protein binding residues. We have different predictors like FMOR-PRED, DSORDP bind, anchor 2. So these all servers are individually available in different links. Okay. So but here in just one point, we have just kept everything for the research community, so that if you wish to find, so instead of showing different web servers, I thought that I should show you in one particular platform where you can see all the web servers, how they are predicting and how the results you can analyze. Then we have RNA binding predictor, which is RDP bind, DNA binding predictor, then linker residue predictor. What are linkers actually? In multi protein domains structure, you have certain link or the spacer which is actually linking. So for that also we have a predictor, and also we have a multifunctional residue predictor, which is actually the moonlighting regions in the protein. So different predictors have been created over here, and by default everything is checked. You can uncheck whatever you wish to take. Suppose I want to know the protein binding, I just keep it here, or else you just put it everything and you can just get whatever things. And one of the most interesting thing of this is that these all are color coded in the sense that the first one is shown gray, then the green one is not very much visible over here, then sky blue, then the pink, and then the violet color. And then you can just click on the run to run the predictions. And there is the help option, which is actually showing how the input file looks like, the material, actually how, because we are using a machine learning approaches, so we are having how do we did the test data set and the training data set, and also we have this benchmark dataset which also any user can download. So this is the dataset that we have actually used for our prediction purpose for running these things. And then the references, and then okay. So let's start with this. So I need to put a protein sequence over here. So I have already downloaded one from this prot, which I am just copying it and I'm pasting over here. Remember one more thing, in Depictor, the minimum length of a protein sequence is around 26 and the maximum is around 5,000 amino acids. So do not exceed your proteins more than 5,000 amino acids, else it will pop up. A pop-up will come and see, it will say that it is not acceptable. Okay, so I am showing you let's say I will start with an example protein. You can just click into this example and it will be showing you a protein sequence. It is a regulatory protein SIR3, and it's this way. This is the one letter code of the protein sequence, and I'm not putting my email ID for the time being, and I selected all these servers and I click the run. So the next page will be coming a Bioinformatics processing page, which is actually will giving you an ID. You do not have to remember this thing. It will automatically come to your email ID if you are giving. So this is again having a story in the sense that this 2020 10 10, if you can see the date today is 2020 10 10, and now it is around 5:12 5 p.m. So that also time is also even like the time, the proper date and the hour and the minutes and seconds. So this is a particular thing which actually is the ID which actually as a unique identifier for any databases. If you are know about this SQL, MySQL or databases, you must be knowing that you need to have a proper key ID for everything. So every details data entry. So here they have given this one. So it is saying that the summation is now being processed. We have to wait, and this is the direct link to the result page. It will come after seeing, but do not refresh this window page manually. If you have any problem, then you can just write to this concerned person. He will just give this query around it. So let's wait for some time till the Depictor is processing the thing. On an average, it takes around 4 to 5 minutes for all the predictors, and for a average protein sequence, which is actually the example one I just tried. I actually wanted to show you the different propensity and the scores. Actually, that's why I just kept running this part. So let it run for a couple of minutes. Until then, if you have any doubts, you can just ask me. Dr. Thakur, if you have any.
I'm
Yeah.
showing you some of the questions.
Okay.
So this is one question from a person.
Okay. So what does IUPRED score and anchor score means to? Okay. Yeah. So IUPRED is a predictor. Anchor is also a predictor. As I was telling, these are the propensity scores. So we have actually in the background, there are number of features have been used to tell that whether this particular amino acid has the chance of being a disordered residue or not. So that score, if it is actually going up higher beyond the threshold level, so let's say I have set my threshold into 0.5, it is 0.6, then that means that score, that means that particular residue has having high chances of being a disordered region. Hope that clears your answers. Anyone else? Depictor is used for what type of analysis? Okay, Depictor actually has a number of predictors. First of all, it is actually predicting the disorder regions and then the functions. What kind of regions which will be having a chances of binding to the proteins or the RNAs or the DNAs, and then the linkers and the multifunctional residues. So if you submit a protein sequence, it will be showing you that this kind of region, this particular region has the high chances of being a disordered regions. That is what we are doing. Yes. In IUPAC 2A score, in IUPAC 2A score is the best score. No, this is not about best score or less score. It is a like wide range of propensity scores. Okay. So when it is crossing the threshold value, so that means that it has the chance of being a disordered region. So you can say that this particular region, like say 26, 30, 31, 32, these residues are having suppose more than 0.5 score. That means that this particular region from 26 to 31, this particular position is having a chance of being an IDP or like IDR. These are disordered regions. Okay. Yes.
Yeah, that much questions only. I can't find
Yeah.
any more questions.
Okay, no problem. Let's see.
And
Okay, the results are ready also. We have the results are ready. That's okay, quite good. So this is the results page. You can just direct link to the page and you can just click into this and it will be showing you the results. Okay. So as I was telling initially, the results page are color coded. So first of all, these are disorder residue as I was showing
we
the gray
can't
color.
view the we can't see the result.
You can see. So what is actually showing? It is showing this.
No, we
Uh,
can't see the result page.
okay, you can see. Just wait a second. I'm again trying to stop the screen and share the screen again. Compare Depictor results page. So is this visible now?
Yes, yes.
Okay,
Yes.
great. So this is how the results page looks like. As I was telling, though there are 6 categories of predictors: the disorder residues, the protein binding, RNA binding, DNA binding, linkers, and the multifunctionals. So these are color coded. So if I hover my mouse over here in disordered, you can see that these are also highlighted. So these are the disordered thing. If I go for the protein, then the green one, this one is highlighted, and the sky, and then this. See, look into this category, look into this part, and you can see this all actually, looks very easy for the biologist people, but when we are writing the course, it actually takes a lot of time for the computational people, because you have to incorporate JavaScript, Java, and many more things to show this kind of color coded and results. Use interesting and like one thing, when you picture is worth more than a text, as they say. So here you can see the color coded. It will be very much useful and interesting to see the results in this format. So by default, you can see that this only binary form is being checked. Okay, here, so zoom is there and you can show help. So one more thing you can do, you can take right click and just you can click on this area and it will be showing, and mouse hovering over here, you can see that this is position 201 to 463. So that means that in the given protein sequence, the residue number 201 to 463 residue is having a disordered region. Okay, and also this one 490 to 509. So in IUPRET and anchor, whatever I was showing in the slides, you did not find this kind of thing. So here what we tried to, we have included IUPRET, anchor, all these servers in a common platform, and to try to uniformity, we have brought the uniformity among the results section. So you can see here the results, the residues. You can just look for any trained biologist also, it will be very much useful. So okay, my protein sequence from 924 to 978 has a protein binding region. Similarly, for the RNA region, DNA region has 201 to 320. It is having high chance of binding to this DNA. So this region will be binding to DNA, and then we have linkers and then also multifunctionals. Yeah, so I am unchecking this one, only binaries, and then you are getting this propensity scores which I was discussing little earlier. So here if you look now, suppose I point this way. So what does it show? This means this is for position is 236 and count is 0.666. So here see, so this is a thing that we have actually incorporated one kind of script. So position means that residue count, residue position. So 236 amino acid position in this protein sequence is actually having a propensity score of 0.666. So this value we have found that when we are running our classifiers or the predictors in the back end, we are getting that this particular amino acid is having a chance of 0.666. So that means it is actually exceeding our threshold value, which is actually 0.5. That means that it is likely to be a disorder. Now let's see, I can hover here. So here if I look position 71, so it is having a propensity score of 0.002. So it's very less. So that means that this position 71, that amino acid over here, it is not having a chance of being a disorder region. So you can see that this particular portion, you can see the propensity scores, it has been like high elevated. And also what we can do, I can just make it a little bigger like this, and also zoom it to this, and just zooming it. So see, you can zoom it also, there is option for zooming, and you can see, look, this 201 position. So I'm starting from this 195th amino acid is having a count of 0.02. So it is not a disorder there. But when we go up and here, see, look a little bit earlier more than here. From here, 201 position is having 0.539. That means that is having a disorder region. So from 201 position to, let's say, whatever it will be showing you. Okay, so individual predictors, individual propensity scores, and the binary scores are shown over here. So all are color coded. So for DNA we have sky blue, for blue, for RNA we have sky blue, anchor green, and the linkers, all the different predictors, it has been shown over here. And here this scale is actually representing the amino acid thing. So we gave a very big sequence, that's why it is actually not showing the amino acid sequence. Also I'm trying to make it more zoom so that it will just show you the amino acid positions name also, but it's not showing here. It will all, so if you take a small sequence, it will actually show. So this is actually, see, this is a very big sequence, so 100 to 200, around 100 residues are over in this case. So it is not able to show the sequence totally completely. But here you can see 90 is Y. Y means tyrosine. Okay, so if I look in zoom here, putting everything, so it will be showing the position. So 115 position is the proline, where my mouse is hovering. 246 is isoleucine, and then the position count, everything you will be finding. So this is a graphical output that the web server is showing you for the common people. So after seeing this visualization only, you can actually have an idea what are my regions and all that. And also it also gives you an option to download the results file in a text file. Now how does the text file looks like, that I will be showing. But before that, here the how this text is being shown, it will be showing you all this very nicely, how this each and every lines in the results looks like. So I'm clicking on the results.txt, it will be giving into another tab. Is the result file being shown? Is the screen, or should I share it again?
No, the result text is not being shown.
Not being shown. Okay, I'm just again changing and sharing the screen once more. Okay, so where is the result.txt? Yeah, here it is. Okay, so this is it's visible now.
Just a minute. Yeah, it's visible.
Yeah. Okay, so this is my protein sequence that I submitted into the Depictor, the highlighted blue one. So greater than P06701 some derivative protein, and this is my sequence. Now, starting from the first one, so let me tell you that in the disordered thing, we had actually taken 3 predictors. IUPRED L, IUPRED short, IUPRED long, IUPRED short, and then the spot. So it might be little confusing for the users. How to use it? There are 3 different predictors. Which one to use? There will be 3 results. Which one is the best one? So for that, to avoid confusion for the user community, what we did, we took all the prediction results from all these 3 servers and tried to make a consensus predictions. So for the first one, what you are seeing is the consensus prediction in that Depictor graphical page also and the result page also. So you can look into this. So what we have given for the disordered, we have given a small d. Okay. So if you look for this, suppose this particular sequence, and also we have used that part kind of letter code so that it is actually taking that much only space. For each and every letter is taking that much space, whatever the dots and the d's are taking. So for example, here this first line is all dots. That means that these are not disordered. Whenever we are standing this d is for disordered. So this means that this is standing from this second row. P, K, Q and all this line is actually a disordered region. Starting from this, you can also get the coordinates from the graphical user, and all here in the text file you can look into this. So this region is actually, so you can see this is actually correlating with this part. So this is actually a disordered region. Then again, this small thing you can just see also and find it out. So easy to use for any like layman also. But like in this particular question, this region is a disordered region. Then this is followed by the propensity scores. So each individual amino acid, suppose let's say the first is M for methionine. Methionine is having a propensity score of 0.016. That means that it is not a disorder. So that's why we are given a dot position. And here from this position we are having, let's say, proline. Proline is actually, if you look again, go for the scores, you will have something somewhere from here around look into here, something from 0.59, 539, 539, all that. So these are the actually disordered regions. So first we are giving the sequence, then we are the binary predictors. Binary in the sense dot or d. Dot for not, d for disorder. Yes. And then the propensity scores for each and individual residues. This is again then the prediction of IUPRED. We again giving the prediction of IUPRED individually also, IUPRED short also, then followed by protein binding. All the individual, everything, the details have been given here, and also the consensus region. So the user people, it will be useful for people to download it and use it for further analysis. Now let me tell you one thing, that in Depictor, again I have to again go to the streamline and share this thing. Okay, tab Depictor, yeah. So here you have the option of selecting any kind of web server. Okay, so if we do not, if you are only interested for disorder residue, select only this one and we'll get the results. Okay, no need to select all the things. This here actually gives you a free and flexible way of choosing any kind of predictors you wish to run. Okay, so it is not like you have to select all the things. So that is the uncheck option is always there. Whatever is your research orientation is, that you want to go for protein prediction or DNA prediction or something, then you can go for that particular predictor, or else any disorder or the moonlight or linker. And oh yes, so that is all about the Depictor. Now moving back to my slides, I will be sharing my slides again. So application window, yeah, we are here somewhere. Yeah, so this is, we discussed about how Depictor works and how the results page looks like, the binary scores, and then the when you uncheck it, you get all the propensity scores as well. So summarizing my today's talk, I say that IDPs or the IDRs, actually these regions actually fail to form a stable 3D structure, but they are very much important for all cellular functions. usually what happens is that IDPs are actually, when they are binding to a proper structured partner, let's say protein or RNA or DNA or anything, linkers or something. So this is how they are functioning. They are undergoing a disorder to order transition. Suppose there is a disorder thumb loop in a RNA and it is interacting with the protein. Because in the disorder thumb loop of a protein, it is interacting with the RNA, then it is getting stable after forming this complex. So this is how this IDPs are actually forming. A lot more is to be needed to learn from these IDPs, which are actually research community is trying to do. And for that, many computational methods are also being developed for the prediction of IDPs or the IDP IDRs and their functional partners. And yeah, we have many more to go. So many things to be learned from these IDPs. Yes. So biology, as I said, is a science of exceptions. We are getting more and more information from the protein communities. So I wish to know more about IDPs in the future. So with this, I'd like to thank you all. And this is my institute where I'm currently working at NIT Durgapur. This is my department. And if you have any queries, you can just write to me in this is my email ID. And yes, that's all for now. Thank you. Thank you all for your time.
Yes.
Yeah,
So that
yes.
was a very informative session, and I'm
Hmm?
very grateful that you took time from your busy schedule to join with us. So I think we have some time, so we will take some questions, and I request all my participants who are viewing us, please post your question in the comment section so that we can take one by one. But remember one thing, that we are having a lot of comments, so sometimes we may we skip some questions. So don't be angry. We'll try to answer as much as possible. So sometimes it may happen that we may have skipped your question. So, but we'll try to cover as much as possible. So here is one question.
Yeah, the examples you gave about IDPs are for eukaryotes. How about prokaryotes? Yes, do prokaryotes do have also, for example, they have also IDPs in their proteome, but so I did not show you. But there are many very good reviews. You can just go to the Google Scholar or ResearchGate. You can just type IDPs in prokaryotes. You can find a number of examples on IDPs in prokaryotes.
Okay, thank you. The next
Although, as I said that in prokaryotes, the number of IDPs are less as compared to eukaryotes. The multicellular organism, the complexity, the things increases. Yeah. So,
yes.
can we use IDP prediction tools for any proteins? Yes. You put the IDP proteins, and if it is not showing any, if it is a structured protein, it won't show you a disordered thing. Yeah, but if they may say, but these are prediction tools, so it might give you some wrong, false negatives as well. So you might get some wrong info also. But the machine learning that we have used, that the tools that we have used are working pretty good and also fast. They have been used in various data sets. For example, the in Depictor we used around 216 disorder sequences and around 216 order sequences taken from the protein data bank. So we are actually taking both the what do you say, the positive and the negative data sets. Okay, so it can be used for any proteins. Yes. On what basis do we set the threshold value for calculating score? Yes, that's for Menka. I guess she's my student. Okay, great. So Menka will discuss about this. The threshold value actually depends on particularly when you are running this algorithms a lot, then you are getting the range, and based on that range, based on that particular thing, again you have to run for the test data set. Whatever you did for the training set, you have to go for the test data set, and then you can just find out that this particular threshold value is actually is the limiting thing from which exceeding which can be a disorder or not. So we can just make that threshold value from that.
Okay, so is another question.
What type of algorithms it follows to score the disorder regions? So as I said, then different predictors are using different kind of algorithms. Some are using propensity score, some are physiological properties, physicochemical properties, some are using this machine learning approaches, but we need to have them authenticate parameters. Okay, the feature selection in machine learning is very important. So first thing is the amino acid composition, which is the first and foremost thing for making any disorder regions. So amino acid compositions, then the evolutionary concept, Shannon entropy, because if you look into the protein structure, the surface residue, as you say, they tend to change, they tend to mutate more as compared to the buried or the inner residue, the interacting partners. So in this interacting regions, they have a low entropy, whereas compared to the surface region, which are having high entropy. So this, suppose entropy can be one parameters. Now solvent accessible surface area. So there are different features which are actually used to make this algorithms. So based on the best features, which we are actually predicting the disorder regions. Yeah. Is it so possible that IDRs will be present in every type of protein? Yes, quite. Yes, that is what IDR are. If you look into protein data bank, look into download a protein sequence, you might be getting some missing residues. So it so happened that people were often confused. Okay, maybe we did a extra crystallography. Sometime there are some missing electron density maps and all that. That's why the residues are missing. But it might so happen that those missing residues are actually the disordered regions. Okay, so that is quite possible that any protein can have a disorder. Yes. Yeah. Any protein that are secreted during stress response of a bacteria can we say is a function of IDP as it activates a certain function or enzyme? Well, no. I would say that no. You cannot. It is not a hard and fast rule that you can say that this is having this function, so I can say this. That this. We need to check. We need to check it. You cannot give it that way. Okay, can we use IDP prediction tools for any proteins, for example, whose structure is available for commercial protein? Yeah, if you have a sequence, if you, because these are all sequence dependent predictors, you need to provide a sequence in FASTA format. Then the tools can be telling you that whether it is a disordered region or not.
Okay. So I think that much questions only. I can't find any more question. So
Okay,
thank you
great.
all. Thank you, Amita, for
Thank you
joining.
so much.
Okay.
Yeah,
So
and it was
hope
very nice. I hope I would be able to make the students understand something on IDPs, which are very much challenging topic as of
it
now.
is a very challenging topic and a very, I would say, has a lot of potential for research also. So
Yeah.
yeah, thank you for your time, and maybe we can see you again, maybe in
Yeah,
some
yeah,
next.
sure, anytime. Yes.
Okay, Amita.
Okay. Okay,
So
great day.
yeah,
Thank you.
so
Okay.
so
I'm
we end
receiving
this.
this.
Yes, so we are ending this session, and thank you all for joining. I just want to tell that we'll share the slides and the email ID of our research persons with you at the end of this session, at the end of our training program. So if you have any question or you are generally interested in some kind of research topic, you can contact them. So this is that's all for today, and we have another session tomorrow. So we have two other speakers tomorrow, and they will be speaking on R, particularly the use of R in statistical analysis of a lot of biological data, and I think that will be a very interesting topic. So I request all of you to join tomorrow, and it is at 3:30 p.m. 3:30 p.m. and we have already shared the link in your previous mail. So check your previous mail. We have the link for the third day, and we'll also share the link tomorrow. Also, maybe tomorrow morning we'll share the link again. So please do join for the last session. I hope that will be a very interesting one. So we're ending this session. Thank you all for joining again. So we are ending this session with this. Thank you.